History of Microbiology, Part 2. Another scientist is Louis Pasteur. In the mid-1800s, he was hired by the French government to take a look at French manufacturing processes and the problem of spoilage, particularly the manufacture of beer. The French are not remembered for their beer. In fact, if we think of beer in Europe, we often think of German beer. German beer, even today, many beer manufacturers have their own proprietary microbiologists and stock cultures of microorganisms to manufacture the beer. What Louis Pasteur found was that the kinds of microorganisms that were being used to manufacture beer and wine in France in the 1800s was different, and that there were spoilage organisms that could enter into those food products and ruin them so that they weren't drinkable. Well, in 1859, when he was studying the spoilage problem, and of course at the time it was thought that spontaneous generation was the cause of the spoilage organisms showing up, he decided, and of course he did not agree with spontaneous generation, but he decided that he could set up a nice, simple but elegant experiment that could demonstrate where the spoilage organisms came from, and at the same time disprove spontaneous generation. What he devised was what we now call is a swan-necked flask. This is an elegant but simple experiment. He had a flask that had a long S-shaped curved opening and neck. The opening essentially was tiny, but still open to the air, so the critics of any previous spontaneous generation procedures could not critique that. And what he knew was that there were tiny bits of dust and microorganisms that were trapped in the bottom curve of this S-shaped neck. Now, as the dust and dirt settles in this trap, this little curve just below the opening of the neck of the flask, if you sterilize the food or the broth liquid, that's a food for microorganisms, in the vessel, in the flask, allow it to slowly cool, and then just don't disturb the flask, that broth will stay sterile, which means no microorganisms present, indefinitely. In fact, some of his original flasks with the open swan neck set for decades, never evaporating, never getting contaminated, or showing growth of an organism. When you somehow dislodge any of the dust and the bacteria that's in that trap just below the opening of the neck, however, you do introduce unwanted microorganisms into that broth, and they will start to grow. And the evidence for the example, or how we determine that there are microorganisms growing, is that the broth gets cloudy. Just as if you were going to take a pot of soup, maybe like chicken soup, let it sit out for days at a time, eventually you'll see it gets really cloudy, and of course, pretty smelly too. Sure enough, his little simple experiment with these open-necked, swan-neck flasks, and you compare them with similar kinds of flasks with the same diameter, but the necks were right straight up and open. The flasks that he used that had straight necks with the tiny opening the same size always showed growth immediately versus the no growth in the broth of the swan neck flask. This is really one of the final procedures or experiments to disprove spontaneous generation once and for all. Louis Pasteur was a prolific, hard-working scientist. His contributions were so numerous, eventually the French government established the Pasteur Institute in Paris, France. But that's getting ahead of the story. In the mid to late 1800s, Pasteur had been studying the process of fermentation, where different types of food products, fruit juices, grains, can be used by microorganisms in a process called fermentation. The microbes use that source of food, and in the process of breaking that food down, using it for nutrition for the microbe, release a waste product, essentially, that's ethanol or alcohol. That's, of course, for us, the drinkable alcohol. That alcohol problem of we want alcohol and not spoilage was one of those problems that Pasteur had been hired to solve, and he did. In the process of solving the problem of spoilage and making sure that food products of various kinds had better shelf life, so to speak, no spoilage, he also came upon a heating process called, today, pasteurization. Of course, he didn't necessarily call it that then, but he found that if he heated food to 56 degrees Celsius for 30 minutes, it was enough to inhibit or kill spoilage organisms so that beer, wine, and eventually dairy products that were also pasteurized had a longer shelf life. Well, we call that shelf life today. But it certainly didn't spoil nearly as quickly as it did prior to this heat process. He also found that gently heating these foods also eliminated disease-causing or pathogenic microorganisms that could also be in the food. Prior to the advent and the more widespread use of this heating process that we now call pasteurization, it wasn't unusual at all for microorganisms that can cause tuberculosis to be transferred to people in those foods, especially dairy products. In the early 20th century, many countries and cities all over the world established pasteurization as a requirement when processing different types of dairy foods, dairy products, to eliminate and prevent the transfer of disease-causing organisms from those foods. Pasteurization does that for us today, even now. But the difference in pasteurization then versus now is that we want to process those foods a little bit more quickly, so the manufacturing process is more efficient. So today, food manufacturers use a flash pasteurization or high-temperature heat pasteurization process. It can be anywhere from about 72 degrees Celsius for 15 seconds, or even slightly higher temperatures for maybe 1 to 2 seconds. In addition to contributing this process of pasteurization that we still use today, something else that we still use today, or that's a legacy from Pasteur, are what I call the second-generation vaccines. The original vaccination procedure was developed by Edward Jenner in the late 1700s, early 1800s. But it wasn't until Pasteur, almost 100 years later, investigated and discovered that if he grew microorganisms that could cause disease, like diseases such as rabies, and he could take sources of the rabies organism and grow it in the laboratory and essentially weaken that organism. So it wasn't nearly as devastating or as what we call pathogenic as if it were transferred from animal to animal or animal to person. That weakened material could be used to essentially provide protection or immunity to an otherwise vulnerable animal or person. Of course, immunity, immunization, vaccination. Pasteur developed the first vaccines to protect a person from rabies. There is no cure for rabies. Instead, we essentially protect ourselves and intervene quickly using the vaccine. These are just a few of the contributions that Pasteur has given us, and part of the reason that the Pasteur Institute was established in honor of his legacy and his brilliance and contributions in the field of microbiology. Another historically important person in microbiology is Rudolf Virchow. Rudolf Virchow proposed that omnis cellula est cellula, all cells from cells, is the fundamental basis of an organism. But if we look at the building blocks of plants and animals and fungi, other kinds of organisms, that the fundamental structure and functioning unit is a cell. And that if somebody's sick, it's actually the cells that are sick inside the body. This is the foundation for our second theory in biology called the theory of biogenesis, that life comes from life, or cells come from cells. It's also going to be a part of a foundation of a third theory in biology called the germ theory, that if we have a disease caused by a microorganism, that microorganism is growing and living, and that the disease is caused by that microorganism, not some mysterious force. Rudolf Virchow, in 1858, realizing that it was the sick cell in your body and the sick cells in your tissues that was the cause of many diseases, reported his findings in 1858 in his text, The Cellular Pathology is Based Upon Physiologic Pathologic Histology. Sick cells, sick tissues. That's why we're sick. In the mid-1800s, a German physician by the name of Robert Koch was studying causes of disease. He was very curious as to why certain diseases occurred, and had an opportunity when he was practicing medicine for a few years in South Africa as a visiting physician, that there were some animals at a neighboring farm who had anthrax, and when he looked at samples of the blood from those animals, he could find tiny little filaments that shouldn't be there. So for the next few months or years, Koch spent plenty of time developing techniques and ways that he could visualize or see these little filaments both in the blood samples and outside of the blood samples. We now call those techniques in vitro techniques. In vitro means outside of the body, usually artificial conditions or growth media. In vivo would mean inside the body. So in vitro techniques, working with those microorganisms under artificial conditions outside the body. He developed techniques for isolating these microorganisms into what we call pure cultures. A pure culture would be a single species. And in the process of trying to figure out ways to get these little organisms separated and easily visualized, he also used a gelling material that one of his friends, Angelina Hess, suggested that he use. It was popular at the time in the early 1800s and mid-1800s for scientists who were studying microorganisms and studying spoilage organisms and disease organisms to use a liquid food that we call broth. It really is just like the soups that we use today for broth, just a clear liquid with nutrients dissolved or suspended in that water. The problem with using a broth culture is you really cannot determine how many individuals are in that sample, and you certainly can't separate them into pure, separate single species. In fact, Louis Pasteur in France preferred to use broth cultures, and today the reason that Pasteur is not remembered for his in vitro techniques is because they relied on broth cultures. They really had a difficult time keeping everything pure and single species and working with those materials versus Koch's work with what we now call solid media, using an agar base, which is a gelling agent, and then mixing different kind of nutrients with that agar so that we can spread our samples or specimens onto the surface of the agar and get nice, well-separated, single, pure species. Robert Koch, working in the 1860s, developed steps that we still use today to determine the causes of infectious diseases. Based on Rudolf Virchow's recommendation that if someone was sick, or if a plant or an animal was sick, that if we took a look at the affected cells in the tissues that appeared abnormal, chances are that's where we would find the cause of disease. So what Robert Koch was able to do was take an animal or person or plant, originally it was an animal, take a specimen from the affected area in the body of that animal, take the specimen and spread it over the surface of an agar, food, because he was looking for some type of microorganism cause of disease. Allow the colonies to form, take a sample of those colonies, spread them on a glass microscope slide, stain them so that we have some contrasting colors, and the stains he originally obtained from some colleagues in Germany, from the stain and dye industries that were developing in the mid-1800s in Germany, and see if there was something sort of unexpected. Like his little filaments that he saw in the sick animals in South Africa that had anthrax, maybe there would be some microorganisms present in the affected tissues or cells of the sick animals, or the sick people, or the sick plants. Once he had well-isolated colonies, so we have what are called pure cultures, single species, or single representatives of the microorganisms in question. Then we're going to take a healthy test subject, expose them to this suspected microorganism that we think might be the cause of the original disease. And what we should see is actually on the next slide. So we took a specimen from a sick animal, plant, or person, got it in pure culture, viewed it through the microscope, saw that we had a single species, took a sample of that pure culture, exposed a test subject to that microorganism, and determined, do we reproduce that same disease in the test animal that we saw in the original sick subject? If we do, if we see the identically same disease, and on subsequent subculture, or taking a sample from the sick test subject, putting it again into culture with agar plates or agar surfaces, and taking a look through the microscope on glass slides, if we find exactly the same colonies, exactly the same species, the second time around from our test subject that we originally saw in our sick individual, that's confirmation that that particular microorganism is the cause of that particular disease. There are two things that we gain from the sequence of steps and procedures. One concept that we obtain from Koch's postulates, which are the steps that are used to determine the causes of what we now call infectious diseases, is that one microbe causes one disease. Technically, when we're working with bacteria and we're spreading out the specimen, we will also find that one bacterium gives rise to one colony. So one microbe, one disease is what we derive from the postulates. One colony from one bacterium is what we discern from the techniques that Robert Koch developed in the mid to late 1800s. Another important physician, Ignaz Semmelweis, actually performed his work about 2 decades before Pasteur and Koch. Ignaz Semmelweis was an obstetrician, and in fact quite a brilliant physician, had his own teaching team of students or interns and residents that he taught, and noticed that in the early 1800s that if you were a new mom and you had a physician as a caretaker after you had your baby, you were 10 times more likely to die of infection than midwives. Midwives had about a 1% mortality rate, and depending upon the resource that you look at from the 1800s, physicians who took care of new moms often had a 10 to 20% mortality rate. Well, at the time it was quite popular if you wanted to be anybody or demonstrate that you had wealth and status, you had a physician take care of you after you had your baby versus a midwife. But, of course, if you wanted to have a higher survival rate, probably a good idea to have a midwife. Regardless, Semmelweis was curious why there was a significant difference in mortality between these two groups. What he noticed, based on a particular kind of disease that would show up, is that there was a disease that we call childbed fever that was occurring in these women who died after childbirth that was very similar to another kind of illness that sometimes happened in men and children called puerperal fever. That's P-E-U-R-P-E-R-A-L, puerperal fever. Puerperal fever and childbed fever has some symptoms that are very common or similar. And in fact, what really helped Semmelweis determine or decide to keep investigating the causes of childbed fever, one of his colleagues, who was a close friend, accidentally cut himself on a scalpel blade during an autopsy of a woman who died of childbed fever. And sure enough, this colleague developed puerperal fever and died. Well, Semmelweis was quite distraught and started to more thoroughly determine what was different about physicians who took care of the new moms versus the midwives. And the most notable thing was that the physicians often would go from an autopsy straight to taking care of their patients, and they might clean their hands off with a cloth, but they might not wash their hands, so that all the material from an autopsy was removed from under their fingernails and their fingers. Even today, it's often difficult to find a clinician who will walk in the room if they're taking care of patients who immediately washes their hands before they touch the patient. In any case, Semmelweis took his interns and his residents and required that his interns and residents wash their hands before touching their patients, and in fact required his interns and residents and himself to use what was called a chloride of lime. And I don't mean limes like the fruit, but the chloride of lime was very much like bleach. It was an antiseptic and disinfectant, some kind of chemical that would eliminate and reduce the number of microorganisms on the hands of the caretakers. Sure enough, when he required hand washing of his team of physicians, he found that he could reduce the mortality rate of women who were being taken care of by physicians to the same mortality rate as that of the midwives. So it really was something that was transferred from the autopsies to the patients. Now, realize the time. This is about at least 10 years before Louis Pasteur started to study microorganisms in great detail. So Semmelweis and scientists at the time in the 1840s still weren't sure that it was microorganisms in the form of bacteria that was causing both puerperal and childbed fever. Another historically important physician is Joseph Lister. Joseph Lister was a famous physician and surgeon from England, mid-1800s, who started to pull all the concepts and ideas together as he read. He read prolifically, he wrote prolifically, and what he read in the 1800s were the writings of Semmelweis, Pasteur, Koch, became aware of concepts like Virchow's concepts of 6 cells, and determined, especially upon reading what Pasteur wrote, that spoilage organisms could cause the decay of food, might be similar to the essentially spoilage organisms that get into wounds, because about half of the patients who underwent surgery in the 1800s would die of post-operative infections. So oftentimes they would say that the surgery was a success, but the patient died. Well, how do you control where these microorganisms come from, and how do you control them in a special little place where you're doing these kinds of surgeries? Well, what Joseph Lister noticed, that there were farmers in the area where he lived and worked that were using a chemical liquid called carbolic acid. Now, carbolic acid is an excellent disinfectant. A disinfectant is a chemical we could use on an inanimate surface to reduce or eliminate microorganism. And at the time, he also used it as an antiseptic, a chemical to be used on skin to reduce or eliminate microorganisms. Now, he didn't know this originally. He just noticed that the farmers were using carbolic acid to control anthrax in their animals. They would pour carbolic acid on the hay, on the wounds of the animals, and sure enough, the animals would get better, and the anthrax didn't spread. The word that we use today to describe the same chemical is phenol, and it's that very sort of medicinal smell that we often associate with hospitals or clinics. In fact, if you go to the store and buy a bottle of just old-fashioned Lysol, if you look on the bottle, one of the chemical ingredients in the original Lysol formulation is a phenol derivative. So, when Joseph Lister realized that the farmers could limit the spread of disease with their carbolic acid, he decided to spray his surgical suites and his patients and everything else he could think of with a fine mist of carbolic acid. Originally, he took the little spray atomizers that were used by women to put their perfumes and spray perfumes, emptied out the perfumes, and instead replaced it with carbolic acid and sprayed the surgical room. He sprayed it on compound fractures. The compound fractures are essentially broken bones that are protruding through the skin. So not only is the bone broken, but there's a wound. And of course, the wounds were the serious problems where infections would occur. He would set the fracture, sew up the wound, spray it with carbolic acid, and the first 3 or 4 patients he performed this procedure on survived. Before the use of carbolic acid, all of those patients were more than likely to die, and certain at least 2 or 3 out of the 4. So once he determined that carbolic acid was a very good means of controlling infection, he started to use it in all of his surgeries. And of course, eventually, we start to use these kinds of disinfectants and antiseptics routinely when taking care of wounds and we're working in surgeries. Now, if you look at the photo on this slide, you should notice several things about this surgical suite, at least the photo at the bottom, that are quite different from what we'd expect a surgical team to look like. For one thing, they certainly don't have any surgical garments. These are just simply frock coats. These are their everyday clothing. They don't even have aprons on to wipe anything off of their hands. They don't have anything covering their mouths. Their hair's uncovered. If you look closely, this looks like somebody's bedroom or some kind of a typical room that we'd see in any kind of household. And in fact, it is. Oftentimes, doctors in the 19th century had their offices and their sites in a house. But the one thing that's different in this picture is that device on the right-hand side that was used to spray the carbolic acid. So even though we don't see the same kinds of appearances, same kinds of techniques that we'd expect to see today in a surgical suite, basically the same kinds of procedures fundamentally were being performed.